This lecture is on muscle tissue. What is muscle tissue? There are three types of muscle tissue: skeletal, cardiac, and smooth. Skeletal muscle tissue is part of the musculoskeletal system. It controls voluntary movement of the skeleton, eyes, and facial skin. Skeletal muscle cells are called muscle fibers because they are cylindrical, multinucleated, and striated. Cardiac muscles is found in the walls of the heart. It controls involuntary movement of the heart. Cardiac muscle cells are branched, uninucleate or binucleated, striated, and joined by gap junctions called intercalated discs. Smooth muscle tissue is found in the walls of hollow organs. It controls involuntary movement of digestive organs and blood vessels. Smooth muscle cells are small, uninucleate, fusiform in shape, and non-striated. This illustration shows skeletal, cardiac, and smooth muscle tissue. Skeletal muscle is attached to the bones, or, in some facial muscles, to the skin. The cell shape of skeletal muscle fibers are single fibers that are very long, cylindrical, with many nuclei. The cell is obviously striated. Cardiac muscle cells are found in the walls of the heart. Their shape and appearance is branching chains of cells. They are uni or binucleate. Uni means one nucleus. Binucleate means two. This illustration down here shows the two nuclei. And they have obvious striations. These dark purple lines are the intercalated discs. Smooth muscle tissue occurs as a single unit muscle in the walls of hollow visceral organs other than the heart. So this is showing the esophagus, stomach, pylorus, and then the small intestine. The single unit muscles are going to contract together. Multi-unit muscles are going to be involved in intrinsic eye muscles, airways, and large arteries. So that means that these will contract at different times. The cell shape is single. Notice it's one cell. Fusiform, meaning pinched at both sides. Uninucleate, so one nucleus, and no striations. The musculoskeletal system contains skeletal muscles and bones that move the body. Cardiac and smooth muscles are found in the cardiovascular system. Smooth muscle is also found in other organ systems, such as the digestive system and the urinary system. This illustration shows the musculoskeletal system with the skeletal muscles attached to the bones. The functions of skeletal muscles include the following: movement of the skeleton, facial expressions, and your eyes, and this movement is voluntary. Maintenance of posture and body position. Muscle tone is the major stabilizing factor of joints. Protection and support of organs in the abdominopelvic cavity. The abdomen is not covered by skeletal bones, so it depends on the musculature for protection. The regulation of materials through sphincters and orifices. So the orifices and sphincters of the gastrointestinal tract and urinary system regulate the ingestion and elimination of materials. Orifices include the mouth, and then sphincters are found along the gastrointestinal tract, such as the cardiac sphincter. And then the sphincters help to retain urine and fecal matter. Heat production results when muscles use energy to move. and this helps to maintain body temperature. I believe as much as 60% of the energy used in work is lost as heat. The characteristics of skeletal muscle fibers include excitability, conductivity, contractility, extensibility, and elasticity. Excitability is the ability to be stimulated by a motor neuron. Conductivity is the ability of an electric signal to travel along the length of the cell membrane. Contractility is the ability to shorten and generate force to move an object. Extensibility is the ability to stretch, and elasticity is the ability to return to original length after shortening or stretching has occurred. Let's look at the gross anatomy of skeletal muscles. Skeletal muscles are organs. Nerve, arteries, and veins serve each skeletal muscle. Skeletal muscles are separated from each other by fascia, and tendons connect skeletal muscles to bones. This illustration shows numerous skeletal muscles found in the arm and forearm. There is a nerve and arteries and veins serving each skeletal muscle. Notice how the nerve branches, as does the arteries. Skeletal muscles will be separated from each other by fascia. Notice the shape of these skeletal muscles. They are also fusiform, meaning pinched at the ends, but they will be surrounded by deep fascia, which is connective tissue that will separate these into individual organs. And then the tendons will connect the skeletal muscles to the bones. And when the skeletal muscles contract, they shorten and pull on the tendons, which then pulls on the bones, and that creates movement. Skeletal muscles contain several layers of connective tissues: the epimysium, perimysium, and the endomysium. The epimysium surrounds the entire muscle and protects the muscles like a leather sheath. It is continuous with tendons. The perimysium surrounds fascicles of muscle fibers. It is a tough fibrous sleeve that protects and supports bundles of muscle fibers. And the endomysium surrounds individual muscle fibers. It electrically insulates skeletal muscle fibers. The epimysium and perimysium are both composed of dense, irregular connective tissue, which is like what is found in the dermis of the skin. And this is what makes these tissues tough and leathery. The endomysium is areolar connective tissue, which is a very delicate connective tissue proper. All three connective tissue layers contain blood vessels, lymphatic vessels, and nerves. This illustration shows the epimysium, the perimysium, and the endomysium. The epimysium surrounds the entire muscle and is continuous with the tendons. It is dense, irregular connective tissue and is tough, like a leather sheath or covering. The perimysium is going to surround fascicles, which are bundles of muscle fibers. The muscle fiber is the name of the muscle cell, that long cylindrical fiber, multinucleated. So a bundle of these muscle fibers is called a fascicle, and it is surrounded by a thick perimysium, which is also made of dense, irregular connective tissue, similar to the epimysium. The muscle fiber or muscle cell is surrounded by the endomysium. This is a areolar connective tissue that is thin and helps to electrically insulate the muscle fibers from one another so that they can conduct impulses individually. If you've ever eaten shredded beef or chicken, the shreds are going to be the fascicles. There are connective tissues that support skeletal muscles, such as tendons and aponeuroses and fascia. So the tendons and aponeuroses connect muscles to bones. Tendons are cord-like, and aponeuroses are sheet-like. They are both composed of dense, regular connective tissue. This is a strong collagenous connective tissue, but the collagen fibers run parallel to one another. And thus, the pull on the bones of the tendons and aponeuroses are what initiate movement when muscles generate force. Fascia describes the collagenous network of membranes deep to the skin. There's deep fascia and superficial fascia. Deep fascia surrounds the epimysium of a skeletal muscle, and it is composed of dense, irregular connective tissue. Superficial fascia is found within the subcutaneous layer of the skin and includes adipose tissue. There's a lot of collagen in our body. This illustration shows the tendons, which are cord-like, and the aponeuroses, which are sheet-like. So these are the dense, regular connective tissues that connect the muscles to the bones. And when they pull on the bones as the muscles contract, this causes the movement of the skeleton. This illustration is showing the superficial fascia as part of the hypodermis, or subcutaneous layer of the integument or skin. And it says it's found between skin and underlying organs. It's composed of areolar connective tissue and adipose connective tissue. The deep fascia is going to be bound to capsules, tendons, and ligaments. So that means it will be bound to the capsules of joints, the tendons of bones, and the ligaments that connect the bones. It is a dense connective tissue made up of collagen and forms a strong fibrous internal framework that basically binds all the organs and tissues together. And then there's even a different layer of fascia, the subserous fascia, between the serous membranes and the deep fascia. All of this is collagen and, of course, the adipose tissue in the superficial fascia. So as I said, there's a lot of collagen in our body. This is showing the fascia making fibrous cords that will connect the integument to the underlying tissues. And these fibrous cords are, genetic, they're inherited, and they're found in females rather than males. And the pockets of adipose tissue that collect between these fibrous cords is what makes the cellulite. So the dimpling comes from the taut nature of the fibrous cords connecting the skin to the underlying muscle. There's actually a surgery that can cut these cords, and that will help reduce the appearance of cellulite. Let's look at the microscopic anatomy of muscle fibers. Muscle fibers are long, cylindrical, multinucleated, striated cells that function to generate force in skeletal muscles. During embryonic development, muscle fibers are formed by the fusion of stem cells called myoblasts. This is why muscle fibers are long, cylindrical, and multinucleated. Cardiac and smooth muscle cells also derive from myoblasts, however, they do not fuse together. Thus, they are smaller and generally uninucleate. This illustration shows skeletal muscle development through the myoblasts. Myoblasts are the stem cells for the muscle tissue. In skeletal muscle, the myoblasts will fuse together. As they fuse together, they will form the immature muscle fiber. This fusion is going to leave behind the nuclei and make the developing muscle fiber a multinucleated cell. The striations will occur from the arrangement of contractile proteins within the muscle fiber. So, muscle fibers contain unique structures. Let's take a look at each one. We'll start with the cell membrane. The sarcolemma is the cell membrane. It is excitable and conducts electrical signals. T-tubules extend from the sarcolemma into the cytoplasm. So essentially they are extensions of the sarcolemma that go into the cytoplasm. A muscle cell's cytoplasm is called the sarcoplasm. The sarcoplasm contains protein rods called myofibrils. The sarcoplasm also contains mitochondria, myoglobin, and glycogen, which are involved in supplying ATP, oxygen, and sugar, respectively. So mitochondria will supply ATP, myoglobin will supply oxygen, glycogen will supply sugar. Notice that the terms sarco and myo are used commonly when describing muscle tissue. Myofibrils are covered in a smooth endoplasmic reticulum called the sarcoplasmic reticulum, abbreviated SR. The sarcoplasmic reticulum stores and releases calcium ions from terminal cisternae. A triad is the union between the terminal cisternae and T-tubules. In this illustration, we see the sarcolemma, which is the plasma membrane of the muscle fiber, the T-tubules, which extend from the sarcolemma deep into the sarcoplasm, which is the cytoplasm. The sarcoplasm is filled with myofibrils, which contain the myofilaments, actin and myosin. Myofibrils are covered in the sarcoplasmic reticula, which is a modified smooth endoplasmic reticulum, and it contains terminal cisternae that join to the T-tubules, forming the triad. Myofibrils are composed of contractile proteins called myofilaments. Myosin proteins make up thick filaments, and actin subunits make up thin myofilaments. Actin is surrounded by the supporting proteins troponin and tropomyosin. The contractile unit of muscle fibers is the sarcomere. The M line anchors thick myofilaments forming the center of the sarcomere, and Z discs anchor thin myofilaments forming the border of the sarcomere. This illustration shows the sarcomeres. The Z discs anchor the thin filaments and form the borders of the sarcomeres. So between two Z discs is the sarcomere. The center of the sarcomere contains the M line, which anchors myosin filaments. The striated patterns of a muscle fiber are formed from the repeating sarcomeres along the myofibrils. Here's the sarcomere illustrated on its own. The Z discs form the border of the sarcomere. The Z discs anchor actin myofilaments. The M line forms the center of the sarcomere. The M line anchors myosin myofilaments. The actin myofilaments contain actin subunits and the accessory proteins tropomyosin and troponin. Tropomyosin has the longer name and is the longer protein. That's how I remember it. And the troponin is going to be the smaller protein. Here we see a sarcomere sliced crosswise along several areas to show the proteins contained at a particular place. Let's look at the Z discs. The thin filaments will be connected to the Z discs. And then in the M line, the thick filaments will be connected to the M line. There are places where the thick filaments do not overlap with the thin filaments. This gives us the striations. And then the A band is where both the thick filaments and the thin filaments overlap, giving us darker striations. Now let's look at the physiology of skeletal muscle fiber contraction. Muscle fibers depend on the excitation from a motor neuron in order to contract. A motor unit contains one motor neuron and all the muscle fibers it innervates. When the motor neuron fires, it stimulates all muscle fibers within the motor unit simultaneously. All of these muscle fibers will contract at the same time. And they are going to be scattered throughout the muscle. This allows for the muscle to generate a weak contraction even when there is just a few signals. So there are small motor units and there are large motor units. Small motor units are easily excited and they are found in the fingers and eyes, so they can twitch very easily. Large motor units are less excitable and they are found in the leg muscles. So it takes a lot more voltage to stimulate these motor units to contract. But when they do, there's going to be many different muscle fibers that contract at one time, and they generate a lot of force and can move powerful muscles found in the leg. This is an illustration of motor units. The motor unit is a motor neuron and all the muscle fibers it innervates. Motor unit number 1 contains a motor neuron and 2 muscle fibers. Motor unit 2 contains a motor neuron and 3 muscle fibers. Whenever these motor neurons excite the motor unit, the skeletal muscle fibers that are contained within that motor unit will contract simultaneously. This micrograph shows a branching axon terminals forming neuromuscular junctions, one per muscle fiber. So this particular motor neuron is connecting to 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 muscle fibers that we can see. So when that motor neuron excites these muscle fibers, they will all contract at one time. What an efficient system. There are 3 main events in muscle contraction: excitation, excitation-contraction coupling, and the cross-bridge cycling. The excitation of the muscle fiber occurs at the neuromuscular junction. Excitation-contraction coupling occurs when an electrical signal travels down the sarcolemma into the T-tubules. This stimulates the release of calcium from the sarcoplasmic reticulum. Cross-bridge cycling occurs when calcium binds to troponin and initiates the sliding of the myofilaments. All right, so event 1, excitation. The neuromuscular junction, abbreviated NMJ, is the site where a motor neuron joins to a muscle fiber. The synaptic knob is the expanded tip of the motor neuron. The motor end plate is the specialized region of the muscle fiber that joins to the synaptic knob. The synaptic cleft is the space between the synaptic knob and the motor end plate. This illustration shows the motor neuron coming in with the neuromuscular junctions. Here's the neuromuscular junction. The motor neuron has the synaptic knob, which is the expanded tip of the neuron. The motor end plate is the specialized region of the muscle fiber that joins with the synaptic knob. You can see here that it's got undulations. And then the synaptic cleft is the space between the synaptic knob and the motor end plate. The neuromuscular junction generally occurs in the center of a muscle fiber. To excite the muscle fiber, the motor neuron sends an electrical signal to the synaptic knob. Voltage-gated calcium channels open on the synaptic knob. Calcium diffuses into the synaptic knob and stimulates the release of the neurotransmitter acetylcholine abbreviated ACH, via exocytosis. Acetylcholine then diffuses across the synaptic cleft and binds to chemical-gated channels on the motor end plate. As the channels open, sodium ions diffuse into the sarcoplasm and brings the resting membrane potential, abbreviated RMP, to a less negative value. An end plate potential, or abbreviated EPP, aka the threshold, is reached when enough sodium ions have flooded in and changed the resting membrane potential from a negative 90 millivolts to a negative 65 millivolts. Here we see in this illustration the electrical signal approaching and reaching the synaptic knob of the motor neuron. The electrical signal will open voltage-gated calcium channels. Calcium will then enter the synaptic knob and bind to vesicles that contain the neurotransmitter acetylcholine. These vesicles will then fuse to the plasma membrane of the synaptic knob, and acetylcholine will be released via exocytosis into the synaptic cleft. The acetylcholine will diffuse across the synaptic cleft and bind to chemical-gated receptors on the motor end plate. These receptors will then open and sodium ions will flood into the sarcoplasm. At rest, the interior of the sarcolemma reaches a resting membrane potential of negative 90 millivolts. The interior of the sarcolemma has a negative charge relative to the exterior. Potassium ion diffusion is the most important factor in establishing the resting membrane potential. Potassium ions diffuse out of leakage channels at a greater rate than sodium ions diffuse in. Large protein anions attracted to the potassium ions also contribute to the resting membrane potential. The leakage of potassium ions out of the cell, coupled with proteins, brings the resting membrane potential to a negative 90 millivolts. The sodium-potassium pump maintains the resting membrane potential at negative 90 millivolts by actively exporting 3 sodium ions and importing 2 potassium ions. This results in a higher concentration of sodium ions outside and a higher concentration of potassium ions inside. This concentration gradient allows for the generation of an action potential, which reverses the resting membrane potential from negative 90 millivolts to a positive 30 millivolts. Sodium ions will rush into the cell during a process called depolarization. The sarcolemma will become positive 30 millivolts during depolarization. Potassium ions will flow out of the cell during repolarization. The sarcolemma will return to a negative 90 millivolts as a result of repolarization. This illustration shows the ionic basis of the resting membrane potential. At rest, the resting membrane potential is going to be a negative 90 millivolts. Voltage is the differences in the charge across the membrane. The primary factor in establishing resting membrane potential is the leakage of potassium ions from the inside of the cell to the outside of the cell. As the potassium ions exit the cell, they leave behind negatively charged proteins, which are large anions. The loss of positively charged potassium ions, along with the remaining negatively charged proteins, bring the interior of the membrane to negative 90 millivolts. The sodium-potassium pump maintains the resting membrane potential by exporting sodiums out of the cell and importing potassiums into the cell. This maintains a concentration gradient between the extracellular fluid and the intracellular fluid or cytoplasm. The gradient will be as follows. There will be more potassium ions on the interior of the cell and more sodium ions on the exterior of the cell. During the action potential, depolarization occurs when voltage-gated, abbreviated VG, sodium ion channels are opened by the threshold value. An influx of sodium ions brings the sarcolemma to a positive 30 millivolts. Repolarization occurs after the depolarization wave passes. Voltage-gated sodium channels close, and voltage-gated potassium channels open. As potassium ions exit, the sarcoplasm hyperpolarizes or becomes more negative than the resting membrane potential. The resting membrane potential will be reestablished by the sodium-potassium pump. This illustration shows a graph depicting the action potential phases of depolarization, repolarization, and hyperpolarization. So at rest, the interior of the cell membrane is a negative 90 millivolts. This is established by the leakage of potassium out of the cell, leaving behind the anions, and it's maintained by the sodium-potassium pump. The cell will remain at rest unless it is stimulated by a motor neuron. This stimulation by the motor neuron will cause the opening of chemically gated channels in the motor end plate. When these channels open, sodium ions will start to flood into the sarcoplasm. This influx of positively charged ions is what makes the interior of the cell membrane less negative. As the interior of the cell membrane becomes less negative, it will approach the threshold value. If it reaches the threshold of negative 65 millivolts, then not only have the chemically gated channels opened, but voltage-gated sodium channels will now begin to open across the sarcolemma. This will result in the depolarization wave that will bring massive influx of sodium ions into the sarcoplasm. And this massive influx of positively charged ions will depolarize our membrane, meaning changing the charge from a negative 65 threshold all the way up to a positive 30 millivolts. Once the depolarization wave has occurred and positive 30 millivolts has been reached, immediately the voltage-gated sodium channels will begin to close. So sodium is no longer coming into the membrane at this time because the channels are closing. Now what is happening is the voltage-gated potassium channels have opened. So that positive 30 millivolts from the depolarization wave closed sodium channels and opened potassium channels. We are no longer getting any positive ions flooding into our sarcoplasm. Rather, we are now seeing the exit of potassium ions out of the cytoplasm. So now we're getting a loss of positive ions from the cytoplasm, and the negative charged anions are going to gather in the area, and we're going to start to repolarize the membrane. So notice we're no longer getting any sodium coming in. Our sodium channels have closed, but we're losing a lot of potassium now. So our membrane is going to become more negative once again. Eventually it's going to hyperpolarize or go below the negative 90 millivolts. So it will become more negative than negative 90 millivolts. This stimulates the voltage-gated potassium channels to close. So now sodium channels are closed and potassium channels are closed. Leakage channels still remain open, and the sodium-potassium pump will work diligently to re-establish the resting membrane potential and bring us back to a negative 90 millivolt state. This is how the electricity of the muscles works. So event 2 is the excitation-contraction coupling. That was all event 1, excitation. So event 2, excitation-contraction coupling. The action potential will travel down the sarcolemma and into the T tubules. Depolarization of the T tubules will open voltage-gated calcium channels on the terminal cisternae of the sarcoplasmic reticulum. Calcium will then flood into the sarcoplasm and bind to troponin. So if you've noticed, so far, the point of all of this is to send an electrical signal down the motor neuron where it's going to open voltage-gated channels. So basically, voltage-gated channels will be shocked open or closed by an electrical signal. So here, on the synaptic knob, we have voltage-gated calcium channels. When the electric signal comes down, it will open the voltage-gated calcium channels. Calcium will enter, bind to these synaptic vesicles, and cause the exocytosis of acetylcholine across the synaptic cleft. Now we're looking at chemically gated channels. The acetylcholine is going to bind to the chemically gated channels. They're named because they respond to the chemical, which in this case is acetylcholine. Acetylcholine will open these channels and sodium will start to flood into the motor end plate. This will bring us up to our threshold of negative 65 millivolts. Once we reach that negative 65 millivolts, we're going to open voltage-gated sodium channels. So these guys are going to be sensitive to electricity, and when they are hit by the appropriate voltage, they will open, and now sodium will flood into the sarcoplasm. And the influx of sodium into the sarcoplasm is going to cause the depolarization of the interior of the sarcoplasm to a positive 30 millivolts. This positive 30 millivolts is then going to open the potassium channels. Notice they are voltage gated as well, so they're going to depend on particular voltage in order to open. As the potassium channels are opening, the sodium channels will close. So we're no longer getting any sodium coming into the sarcoplasm, but now we're going to lose potassium. And as we lose potassium, we're going to leave behind negatively charged protein anions, and we're going to repolarize our membrane, bringing us back into the negative values. So this depolarization wave is going to continue all the way down the sarcolemma, where we're going to have depolarization followed by repolarization. The T tubules will take the action potential, which is the depolarization followed by repolarization, down into the muscle fiber, into the sarcoplasm. Along the T tubules will be more voltage-gated channels, which are called the calcium channels. So this is the second place that we have seen calcium channels thus far. When the voltage-gated calcium channels open, the calcium ions will then diffuse out of the terminal cisternae of the triad into the sarcoplasm. The calcium ions will then go and bind to troponin, which is located on the actin subunits. So when calcium binds to troponin, it causes troponin to change shape. As a result, the blocking action of tropomyosin on actin is removed. With actin exposed, myosin can attach and form cross bridges. The cross bridge formation enables myosin to slide actin towards the center of the sarcomere. This step couples excitation via the motor neuron at the neuromuscular junction with the contraction of the sarcomere through the sliding of the myofilaments. So this illustration shows the calcium ions that are come in and will bind to troponin. Troponin will then change shape, and as it changes shape, it bends laterally, basically, and moves troponin tropomyosin away from the binding sites. Tropo in this case means in the way. So troponin and tropomyosin are in the way of myosin. So when calcium binds to troponin, changes shape of troponin, and moves tropomyosin out of the way. Now myosin can form a cross bridge, which is the attachment of the myosin to the actin subunit. So event 3 is the cross bridge cycling. Cross bridge formation occurs when myosin heads attach to actin. At this moment, myosin has an ADP molecule and an inorganic phosphate attached to its head. The power stroke occurs when the ADP and inorganic phosphate are released from myosin. This causes the myosin head to pivot, sliding the actin myofilament towards the M line, which is in the center of the sarcomere. Detachment of the cross bridge occurs when a new ATP molecule attaches to the myosin head. Reactivation of the myosin heads results from the hydrolysis of ATP to ADP and inorganic phosphate. This illustration shows the cross-bridge cycling. In the first step of cross-bridge cycling, the calcium ions must bind to troponin, changing the shape and then moving tropomyosin away from the binding sites. The binding sites are now exposed to myosin, my apologies. Myosin is in the activated state at this time. Once calcium binds to troponin and the tropomyosin no longer covers the myosin binding site on actin, cross-bridge formation can occur. So myosin heads, which are already in the cocked position or active position, bind to the exposed myosin binding site of actin. This connection is called the cross bridge. So at this moment, the myosin head has an adenosine diphosphate molecule, or ADP, and an inorganic phosphate attached to it. And the next step is the power stroke. The ADP and the inorganic phosphate are released from the myosin head. This causes the myosin head to pivot or swivel towards the center of the sarcomere, pulling the attached thin filament with it. This is called the power stroke, and the actin is going to slide towards the center of the sarcomere. Now the cross bridge must be broken. In order to do that, ATP binds to the ATP binding site of the myosin head. This causes the release of myosin from the binding site of actin. The cross bridge is now broken. It's time to re-energize the myosin head so it can form another cross bridge on another area of that actin. This occurs when ATP is split into adenosine diphosphate and an inorganic phosphate. The energy that is released during this hydrolysis re-energizes the myosin head. It is now cocked in the ready position again and can form a new cross bridge with another area of the actin so that it can continue to slide that actin towards the center of the sarcomere. So eventually the actin molecules or myofilaments will fill in the space, the potential space of the M line. The Z discs are now closer together. Recall, the Z discs make the border of the sarcomere. The sliding filament theory states that the sarcomere shortens as a result of the sliding of thin filaments past the thick filaments during cross-bridge cycling. So this illustration shows a relaxed skeletal muscle in figure A. Notice that the Z discs are farther apart from each other and there is a potential space within the M line that's called the H zone. During the sliding filament theory of contraction, the actin molecules have slid towards the center of the sarcomere as a result of the cross-bridge cycling. And now the H zone has disappeared because the actin is filling it in, and the Z discs are now closer together. For a muscle to return to its resting state, a motor neuron no longer sends a signal. The axon terminal stops releasing acetylcholine via exocytosis. Acetylcholine is removed from the synaptic cleft via the enzyme acetylcholinesterase. Chemical-gated channels close on the motor end plate. Voltage-gated sodium and potassium channels close. The sarcolemma and the T-tubules return to resting membrane potential. Calcium ions are actively pumped back into the sarcoplasmic reticulum. Tropomyosin returns to position covering the actin, and the muscle fiber's elasticity returns it to its resting length. Rigor mortis occurs around 3 hours after death. This is because the ATP levels in the skeletal muscle are exhausted. Calcium is unable to be pumped back into the sarcoplasmic reticulum, and thus calcium ions remain attached to troponin. Myosin remains in cross-bridge formation because there is no ATP to break the cross-bridge. And rigor mortis will continue for 15 to 24 hours post-mortem until proteins begin to decompose and the muscle tissue breaks down. These videos will be linked so that you can watch them to help you visualize the 3 major events of muscle contraction. The events at the neuromuscular junction, excitation-contraction coupling, and cross-bridge cycling. Muscular dystrophy is a group of inherited muscle-destroying diseases. Duchenne muscular dystrophy, abbreviated DMD, is the most common and severe type. It is an X-linked disease, which means it will be expressed in males if they have the gene on their X chromosome. This means it is a recessive trait. And so if the recessive trait is on the X chromosome, then it will be expressed in the males because they do not have another X with a dominant trait to mask it. Females who have the trait that is masked will not have Duchenne muscular dystrophy, but they will be carriers. Dystrophin is a protein that connects to the cytoskeleton of the muscle fiber and helps stabilize the sarcolemma. Without dystrophin, the sarcolemma tears during contraction. Damaged muscle fibers will undergo apoptosis or programmed cell death as a result of the damage. There is no cure for Duchenne muscular dystrophy. However, the steroid prednisone is the only drug that improves muscle strength and function. This illustration shows dystrophin. In a normal muscle tissue, we can see a healthy amount of dystrophin connecting the cytoskeleton of the muscle fibers. In the affective muscle tissue, we see the dystrophin breaking down. This will cause the cells to atrophy as they undergo apoptosis. The muscles will atrophy and lose mass. Myasthenia gravis is a disease characterized by generalized muscle weakness. It involves a shortage of acetylcholine receptors. It is an autoimmune disease, and the receptors are destroyed as the disease progresses. Symptoms include a droopy eyelid and mouth, difficulty swallowing, double vision, and unsteady walk. It is a weakness and rapid fatigue of muscles under voluntary control due to the loss of acetylcholine receptors. This is a healthy muscular cell with the motor end plate. This is a diseased motor end plate and a loss of acetylcholine receptors in a patient with myasthenia gravis. Without the acetylcholine receptors, the acetylcholine doesn't have anywhere to bind on the motor end plate, and these chemically gated channels will not open, sodium ions will not go into the membrane. The threshold will not be reached, and the action potential will not be generated. Tetanus results in spastic paralysis caused by toxins from the bacterium Clostridium tetani. The toxin blocks the release of inhibitory neurotransmitters in the spinal cord, resulting in the overstimulation of muscles. Vaccinations prevent this life-threatening condition. Botulism results in muscular paralysis caused by toxin from the bacterium Clostridium botulinum. This toxin prevents the release of acetylcholine at the synaptic knobs. Although toxin ingestion can be life-threatening, careful injections of it can treat spasticity due to cerebral palsy, migraines, or can be used for cosmetic purposes, such as to help diminish wrinkles. That's because it paralyzes the muscles of the face that are connected to the skin, and that paralysis, that temporary paralysis, decreases the muscle tension pulling on the skin, and that's what actually is causing the wrinkles, is the pulling of the muscle on the skin. And that's why Botox will diminish wrinkles. All right, so let's look at the energy for skeletal muscle contraction. Muscles use a lot of energy during contraction. ATP is used for the following: cross-bridge cycling, and then pumping the calcium ions back into the terminal cisternae of the sarcoplasmic reticulum when the muscle fiber is returning to rest. Approximately 10 million molecules of ATP are used per muscle per second during contraction. In order to keep up with such great demand, ATP must be regenerated as fast as it is used. ATP reserves can only provide about 4 to 6 seconds worth of energy. This is the ATP cycling diagram, as we learned earlier. When ATP is hydrolyzed into ADP and inorganic phosphate, energy is released for cellular work. In order to regenerate ATP, energy from food is needed, and the mitochondria is able to use this to generate the most ATP. So ATP regeneration occurs in 3 ways: direct phosphorylation, anaerobic respiration, and aerobic respiration. During direct phosphorylation, creatine phosphate is used and can power contraction for up to 15 seconds. During anaerobic respiration, glycolysis is used to regenerate ATP and can power contraction for up to 1 minute. Glycolysis is the breakdown of glucose, and it occurs in the cytoplasm. The mitochondria is not needed for anaerobic respiration. Aerobic respiration generates ATP from cellular respiration in the mitochondria, and it can power contraction for hours. So here we see that the creatine phosphate can power contraction for up to 15 seconds, glycolysis for up to a minute, and then the oxidative system uses oxygen and breaks apart glucose and fatty acids further than the glycolysis will, so it oxidates these molecules and produces an indefinite supply of ATP. As long as there's, plenty of oxygen available, plenty of energy nutrients available, plenty of mitochondria available, then we can continue to regenerate ATP. So this is basically what keeps you alive, is the aerobic respiration. That's why we need to breathe oxygen. So let's look at each of these in a little more depth. Direct phosphorylation uses creatine kinase to remove a phosphate from creatine phosphate and attach it to an ADP molecule. Muscle cells can store up to 3 times more creatine phosphate than ATP. Only 1 ATP molecule can be produced per creatine phosphate, and so this is a very quick method of producing ATP, but it can only last us about 15 seconds. So this illustration shows the creatine kinase taking the phosphate from the creatine phosphate and transferring it over to an adenosine diphosphate, and this phosphorylates the ADP, returning it back into a new ATP molecule. And now we have creatine here, which will then be joined with another phosphate so that the cycle can continue. Anaerobic respiration, also called glycolysis, does not require oxygen or blood flow. During glycolysis, a molecule of glucose is split into two molecules of pyruvic acid. This generates two molecules of ATP and is useful for quick, intense bursts of energy. Lactic acid is a byproduct and can result in muscle burn. It's called lactic acid fermentation, and it lowers the pH of the muscle fibers. This illustration shows that glucose from glycogen breakdown or delivered directly from the blood sugar is going to undergo glycolysis, which is the breaking apart of the glucose into two molecules of pyruvic acid. So we already generate 2 ATP. If there's no oxygen present, then the pyruvic acid will be fermented into lactic acid and released into the bloodstream. If oxygen is present, then the aerobic pathway will begin. So aerobic respiration, also called cellular respiration, requires oxygen and blood flow. It occurs in the mitochondria. It utilizes pyruvic acids from the glycolysis, fatty acids, and amino acids. Peroxisomes will prepare the fatty acids and the amino acids for use in the mitochondria. Cellular respiration produces 32 to 34 ATP molecules per glucose molecule, and it will vary for fatty acids and amino acids. There's so much ATP produced that this generates 95% of the ATP required for resting or moderate activity. So in this illustration, we see that we have gone through the process of glycolysis and have formed the pyruvic acids. We've gotten our 2 ATPs already. But if oxygen is present, then the pyruvic acid can be further oxidized and then enter the mitochondria, where it will undergo aerobic respiration. As a result, 32 ATP molecules can be produced. So add those with what's already made from glycolysis, and we can get 32 to 34 ATP per glucose molecule. Fatty acids and amino acids will be processed by the peroxisomes, and then they will be sent to the mitochondria for cellular respiration. So this illustration shows that within the first few seconds of short duration exercise, our ATP stores are used first, followed by the creatine phosphate phosphorylation of ADP into ATP, and then glycolysis. So we've sprinted for about a minute there. Prolonged exercise will require aerobic respiration, and ATP will be generated by the breakdown of several nutrient energies and will be fueled by the aerobic pathway. And this will use the oxygen released from myoglobin or delivered directly from the blood, and the oxygen deficit will be paid back as a result. So we'll get to the oxygen deficit in a second, but let's talk about aerobic endurance and fitness first. So aerobic endurance is the length of time a muscle can continue to contract using cellular respiration. Aerobic fitness increases the ability of the heart to pump blood efficiently, the lungs to exchange gases, such as, carbon dioxide and oxygen, increased myoglobin and glycogen stores, so we store more oxygen in our muscles and more sugars, and it increases the burning of fatty acids for fuels. So that's why you've heard that having a lot of muscles will help you to burn more fat. So aerobic fitness increases your aerobic endurance. It also increases your cardiovascular fitness, so the ability of your heart to pump the blood, as you can see, and the lungs to exchange gases. So it's a win-win all around. The oxygen debt is the extra amount of oxygen that the body must use for restorative processes. So in the muscle fibers, oxygen will be used to restore myoglobin and glycogen levels, regenerate ATP and CP or creatine phosphate, and repair torn connective tissues. In the liver, oxygen is used to convert lactic acid back to glycogen and to restore glycogen levels. This is why breathing rate is increased after strenuous activity. Aerobic fitness makes oxygen debt more efficient, and you'll find that you don't have to stop and catch your breath as much, and you can go for a further endurance. Humans generally only use about 25% of the oxygen inhaled. The rest is waste. Now let's look at skeletal muscle fiber types. Muscle fiber types are categorized by power, speed, and duration of contraction. Power is related to the diameter of the muscle fiber. Speed of contraction refers to how fast the variant of myosin ATPase can hydrolyze ATP. And the duration of contraction refers to how quickly the action potential is propagated and the muscle returns to rest. So for power, large muscle fibers have more myofibrils and are thus more powerful than smaller muscle fibers. For speed, slow twitch fibers will hydrolyze ATP slower than fast twitch fibers. For the duration of contraction, fast fibers have a quick action potential and reuptake of calcium into the terminal cisternae, meaning they return to rest quicker than slow fibers. Muscle fiber types are also characterized by their method of regenerating ATP. Oxidative fibers regenerate ATP using cellular respiration. Glycolytic fibers regenerate ATP using glycolysis. Since oxidative fibers use cellular respiration, they have an extensive capillary network, an abundant myoglobin and mitochondria, and this gives them a red appearance, and they are fatigue resistant. Since glycolytic fibers use glycolysis, they have a less extensive capillary network. They have fewer myoglobin molecules in mitochondria, and this gives them a white appearance because of the lack of blood flow. And thus, they are prone to fatigue. However, there are benefits to having both these types of fibers. Let's look at the three types of fibers based on whether they are slow or fast, and whether they are oxidative or glycolytic. Type 1 fibers are called slow oxidative fibers, and they are best suited to maintaining posture and endurance activities. They are half the diameter of other fibers. They contract slowly with relatively little power. They use cellular respiration, making them fatigue resistant and able to contract for long periods of time. And they have rich capillary supply and a red color due to the abundant supply of myoglobin. Type 2A fibers are called fast oxidative fibers. They are the least abundant fiber type, but they are useful for walking. They are intermediate-sized fibers that have quick and powerful contractions. They use cellular respiration, but they have fewer capillaries, myoglobin, and mitochondria than type 1 fibers. Therefore, they have a pink color due to the lower supply of myoglobin. Fast glycolytic fibers are 2B fibers. They are the most abundant fiber type, and they are best suited for short-term, rapid, and intense movements. These fibers are the largest in diameter, have quick and powerful contractions, and they fatigue quickly because they use glycolysis, and so they're not using oxygen. Thus, they have few mitochondria and a low capillary supply, which gives them a white color. This illustration shows the three different types of muscle fibers. The type 1 fibers, or slow oxidative fibers, are red in color due to their large capillary supply, and thus they are also fatigue resistant and have a high endurance. Fast oxidative fibers are type 2A fibers. They are pink in color because they still undergo some cellular respiration, However, they have less mitochondria and capillary supply. They can contract quicker than slow oxidative fibers, but they're more quickly to fatigue. And so these are the least abundant fiber type, and they are useful in walking, which of course is very important. Then we have the fast glycolytic fibers, which I was shocked to learn that those are the most important or the most abundant fiber type. But they have the power, but are also quick to fatigue because they're thick but have less blood supply, and they have really quick and powerful contractions. Most muscles have a mixture of fibers. This gives them a range of contractile speeds and fatigue resistance. However, all muscle fibers of a particular motor unit are of the same type. Muscle fiber types are genetic, but some can be modified by exercise. Marathon runners have a high percentage of type 1 fibers in their lower limbs. Sprinters and weightlifters contain a higher percentage of type 2B fibers. Type 2B fibers can temporarily convert to type 2A fibers with endurance training. Okay, now let's talk about some physiology of skeletal muscle tension. Muscle tension is the contractile force generated to move an object. A skeletal muscle provides the effort to move the resistance. Resistance is provided by the bones, flesh, and any added resistance. Muscle tone refers to the fact that even when skeletal muscles are relaxed, they are almost always slightly contracted. This keeps the muscles firm, healthy, and ready to respond to stimulation. It also stabilizes joints and maintains posture. Here we can see some muscle movement generating force. The bone is going to act as the lever, the joint is the fulcrum, and the muscle is the effort. So the resistance is going to be your flesh and whatever you are holding. Muscle contractions exhibit the all or none law, which states that it must reach the threshold value in order to contract. So remember, that's the threshold value of -65 millivolts. This dotted line indicates the threshold value. So if our stimulus is under the threshold value, there will be no response. A muscle twitch is one muscle contraction resulting from a single electrical stimulus from a motor neuron. This is generally illustrated in a lab, and you can measure, using a myograph, 3 periods of the muscle twitch: the latent period, the contraction period, and the relaxation period. The latent period is the time after the stimulus but before the contraction begins. It is the time needed for the cross-bridge cycling to begin. So it's the time needed for excitation to occur and excitation-contraction coupling to occur. So once the cross-bridge cycling begins, then we have the contraction period. This is the time when tension is increasing and the cross-bridge cycles are engaged. So the myofilaments are sliding across one another. The relaxation period is the time when the tension is decreasing back to the baseline. It generally lasts a little longer than the contraction period. Here's the myograph showing the muscle twitch, and we see the latent period. So there has been the electric stimulus coming in, and this is in milliseconds, so it's still very short. And then once the excitation contraction coupling has occurred and cross-bridge cycling is engaged, the contraction period begins. And we see that the relaxation period is longer than the contraction period. And this is the time that it takes for the tension to decrease back to baseline and for all those events for a muscle returning to rest to take place. So the frequency of stimulation increases when the brain fires many nerve impulses to the muscle fibers. A higher frequency of nerve impulses results in muscle fibers not having time to fully relax. This results in a process called wave summation. So wave summation adds the force of each contraction together when a muscle fiber is continuously stimulated, and this results in stronger contractions. This can lead to tetanus. Incomplete tetanus is sustained contraction that occurs when the waves are closer together and continue to strengthen. Complete tetanus occurs when the waves are fused. This is seen only in the laboratory because muscles cannot respond to greater than 25 stimuli per second. So here we see the increased frequency where it's less than 10 stimuli per second and the muscle can go back to resting in between stimuli. So this is the muscle twitch. So move, relax, move, relax, move, relax. But if you're holding something like heavy, or you're holding a position, doing yoga or whatever, and you're in place, you're going to do what's called wave summation. And so the brain's going to continue to send frequencies to your muscles, causing them to hold their positions, and they're not able to relax. And so this increased frequency is going to actually cause these muscle contractions to summate or add together, and the result is that they are going to increase their force until finally they give out once they're fatigued. Incomplete tetany is when we see the sustained contraction that occurs when the waves are closer together and continue to strengthen. And then here we see that above 25 stimuli per second, we get into this realm of tetany, which can only occur in the laboratory, and the contractions are completely fused together. So basically, think about the most intense seizures, And then eventually all the materials will be exhausted. ATP, glycogen stores, oxygen, etc. And fatigue would occur. Motor unit recruitment. So remember, the motor unit is a motor neuron and all the muscle fibers that innervate. Motor unit recruitment occurs when a muscle experiences strain and the brain increases voltage. And this increased voltage will result in the recruitment of larger motor units. So this is getting into like electrical engineering stuff here. And so this increased voltage will also result in a stronger contractile force, because we're getting larger motor units waking up and engaging stronger contractions in our leg muscles, our arm muscles. So motor units, if you remember, vary in their sensitivity to stimuli. Small motor units are more responsive than larger motor units, and thus they are recruited first. And this gives you the twitch and things like that that are very fast, like in your fingers and your eyes and your mouth when you're talking. Larger motor units require higher voltage to be stimulated. So lifting up something heavy or standing up, things like that, your brain's going to send a lot of voltage to those muscle fibers in order to wake them up. So this is showing the increments in the voltage. So by each increment of our brain sending a higher voltage to our motor units, we end up seeing that, at a certain point we get maximum contractions where all the motor units have been engaged. That's generally, of course, not going to be the case. It'll be, around here and here. So this is motor unit 1 recruited first, and then here we see motor unit 2 with the medium fibers secondly, and then our large fibers are recruited last. We have two different kinds of contractions, those that are able to actually move an object and those that cannot. So isometric is like equal force. And so this occurs when a skeletal muscle is contracting, but the force is not enough to move an object. So it's basically like equal to the object that it's trying to move, or weaker if you would. So it occurs when holding a heavy object in place, or when pushing or pulling a heavy object without moving it. Isometric contractions are used when shoveling snow or things like that. And this can increase a person's blood pressure to dangerous levels. And that's why a lot of times people will have heart attacks when they are out there shoveling snow. Because that isometric contraction occurs when you're holding the shovel full of snow and then moving it from one place to the other because you're holding it steady. Isotonic contractions occur when a skeletal muscle is contracting and the force is great enough to move an object. So this is generally what occurs when we are able to lift things and move things and push things and pull things. So here we see a man holding his baby, and that is going to illustrate isometric contraction when the skeletal muscle tension is less than the resistance. Although tension is generated, the muscle does not shorten and no movement occurs. So holding that baby in place. But if he's lowering and raising the baby in space using, the flexion and extension of his muscles, then he's doing isotonic contractions. And so when he's flexing his muscles, he's doing concentric contractions, and that's bringing the baby forward towards his body. And then in eccentric muscle contractions, he's extending. So remember that, eccentric, extend. So he's lengthening his muscle and he's extending his arm and lowering the baby down. The length tension relationship states that when sarcomeres can produce maximal tension if the thick and thin filaments overlap between 80% to 120% during rest. So this is their resting length. So basically they have this optimal overlap that they will have when they're at rest in order for cross-bridge cycles to form once the excitation and the excitation contraction coupling has occurred. Muscle fatigue is a reduced ability or inability of the skeletal muscle to produce muscle tension. And this is pretty rare, but it can happen. The primary cause is the depletion of glycogen stores. You just run out of fuel, run out of gas. Other causes may be seen at the following events. So there can be excitation of the neuromuscular junction may not occur due to the presence of insufficient calcium ion stores or a decreased number of vesicles to secrete the acetylcholine, or as we saw earlier, myasthenia gravis, where we don't even have the receptors for acetylcholine on the motor end plate. Excitation contraction coupling may not occur if there is an imbalance in electrolyte stores, like the sodium ions and potassium ions. Cross-bridge cycling may not occur if there is an increased level of inorganic phosphate. An increased level of inorganic phosphate will interfere with the inorganic phosphate released from the myosin heads, and this will slow contraction. Because remember, when the ADP and the inorganic phosphate is released from the myosin head, that's what causes the myosin head to pivot forward, sliding the actin towards the center of the sarcomere. And so if that's not able to happen, then the myosin cannot pivot towards the center of the sarcomere. Inorganic phosphate may also bind with calcium ions and interfere with the function of troponin. So it'll make calcium phosphate. And then the calcium is not able to bind to troponin. Remember, troponin changes shape and moves tropomyosin away from the active sites on the actin, and that exposes the actin to the myosin, so that cross-bridge cycling can begin. All right, the last section is on the exercise and the aging processes of the skeletal muscles. Skeletal muscles will change in appearance and performance in response to physical training. So endurance exercise uses type 1 fibers. Slow oxidative fibers become more efficient as a result by producing more mitochondria, utilizing glucose and oxygen more efficiently, and burning fatty acids for fuel. So type 1 fibers are endurance fibers, and they can increase your aerobic endurance, and they can also increase strength somewhat. Like you can become quicker at your swimming stroke or walking, but they don't increase in size. So you will increase your strength, but you won't increase bulk. Resistant exercise uses type 2B fibers, which are the fast glycolytic fibers. And this is what will cause muscle hypertrophy. So muscle hypertrophy will occur, which is the increased fiber size from an increase in myofibrils, connective tissue, or both. So folks will do high intensity and low intensity exercises, doing resistance training. High-intensity exercises will increase the size of the myofibrils. So it will actually add more proteins, the more actin and myosin, into your myofibrils. Low intensity will increase the size of the connective tissue. So you'll be adding more collagen fibers to the paramysium and such. So this both together will result in larger and stronger muscles and tendons along with increased bone density. So that's win-win all around as well. This illustration says you need both myofibrillar hypertrophy and sarcoplasmic hypertrophy to get big fast. So in A, it shows high intensity and low volume. So that means that you're pumping weights really fast, but you don't have a really heavy weight in your hand. And so that's going to give you myofibrillar hypertrophy. So that's going to increase the size of your contractile proteins. And then to do the low intensity and high volume, you're going to go really slowly. That's a low intensity, but you're going to have a heavy weight. That's when you see those guys like with really big dumbbells and things like that. They're building up their sarcoplasmic hypertrophy, so basically their connective tissues. The overflow or the overload principle states that forcing a muscle to work hard promotes increased muscle strength and endurance. So muscles are going to adapt to increased demands, and they must continue to be overloaded to produce further gains. Otherwise, they will plateau. That's why, cross-training and things like that are so important, because your muscles are highly adaptive, and as they increase in their fitness and you're able to perform these exercises with ease, the muscles adapt and they plateau without any further gains. Sarcopenia is a condition that occurs as a person ages, and muscle mass will naturally atrophy and be replaced by connective tissues. In this case, it'll be the subcutaneous fascia or superficial fascia, like adipose tissue. Muscles will also atrophy from a lack of exercise. And when muscle fibers die, they are replaced by connective tissue and fat. So although sarcopenia is unavoidable completely, it can be slowed through exercise. Slow oxidative fibers tend to predominate in older people. So here we see this incredible woman named Ernestine Shepard. She is an American bodybuilder who is best known for being at one point the oldest competitive female bodybuilder in the world, as declared by the Guinness Book of World Records in 2010. As of 2019, she's 82 years old, and she's still active, albeit no longer competitive bodybuilder. So she's 82 years old in this picture. Just let that soak in for a minute. So hopefully she can be an inspiration to us all. I believe she started to do her weightlifting when she was in her 50s. She and her sister, and I hope I get the story right, but she and her sister had made a pact to get healthy together, and then her sister became ill and passed away. And so as a result of that, Ernestine did not give up on her goals. She wanted to, but she persevered for the memory of her sister, and that's when she ended up being a competitive bodybuilder. And the comic states, the handle on your recliner does not qualify as an exercise machine. Thank you all. Be blessed.